It is with great pleasure I introduce Phil Stevenson, who's going to talk about sex, drugs and pest control, ecology and application of plant chemistry.
Thank you very much, Andrew. So it's a great privilege and a great honour for me to be speaking to you today as Professor Phil Stevenson, Professor of Plant Chemistry. It's a great honour to have this awarded title from the University of Greenwich. Some of you may be here because I've used an unashamedly sensational title, and of course this has been on purpose, and it's partly to do with the fact that I'm very conscious of the fact that I did have to talk about chemistry, and I've got chemistry in the title. And we are generally pretty chemophobic people. I mean, I'm a chemophobe and I'm an analytical chemist. So I've tried to disguise the word chemistry, but it has puzzled me as to why chemistry does have such a bad name. Obviously there have been some events throughout our history that have not helped chemistry, DDT and mustard gas to name two. But actually I would argue that chemistry is less dangerous than biology and physics. If you consider the last time that you were actually off sick from work, I would hazard a guess it was either to do with a biological organism or to do with a combination of force, mass and gravity that might have left you with some accident, and in fact I've had one of these. And in fact it was probably chemistry that you took to cure the symptoms of those illnesses. So why do biology and physics get such better press than chemistry? They obviously have great champions, but it's hard to know which came first. Our passion for wildlife, or the BBC's passion for making wildlife programmes. Even physics, amazingly, has now become really cool and funny, as Professor Brian Cox and a whole host of comedians are all celebrating physics and planets and space. Yet the great exponents at the moment of chemistry appear to be somewhat different. This is Walter White, the gun-toting, pant-wearing former schoolteacher of Albuquerque, who's now synthesising crystal meth in the deserts of New Mexico. And even when we try and give chemistry a good impression, we get someone like Dr. Peter Wathers, who delivered a Christmas Royal Institution lecture and really confirmed everyone's suspicion that chemists are simply raving pyromaniacs. So, as a chemist, it's my duty today to give you a story about chemistry that shows that chemicals and chemistry can be safe and still be fascinating. I'm going to have a particular emphasis on plant chemicals. I'm going to use examples from my own work, but also work from my collaborators who I'm working with at the moment. I'm afraid there will be no explosions, but there will be some sex and there will be some drugs, but perhaps not quite the ones you were expecting. So there's no question that plants are quite extraordinary chemists. We know of somewhere in the region of 50,000 different natural product structures from plants, and there are estimated to be perhaps somewhere in the region of 100,000 plus. We find them every year. And that's not bad from about 300,000 species of plants. But what are they for? Well, for us, of course, we have many uses for plant chemicals. In fact, it's argued that somewhere in the region of 50% of the best-selling pharmaceuticals on the planet are either from plants or fungi or are derivatives of those compounds. And I've got some examples on the screen for you now. We've got cancer drugs, we've got analgesics, we've got even caffeine, which I'm sure many of you have taken today, as a subject about which I'll speak a little bit more later. But it's what the plants are produced in the plant for, it's what the chemicals are produced in the plant for that's most fascinated me. And of course, we see plant chemicals in plants as colours primarily, and this is for many reasons. Of course, chlorophyll, the molecule that makes plants look green, is a very important molecule, of course, because it enables the plant to harness the energy from sunlight and convert carbon dioxide to oxygen, and in the process make sugar for the plant, arguably the most important molecule that plants are producing, for us at least. And of course we get colours for attracting pollinators, and we get colours that attract herbivores when fruit is ripe, so that they can distribute the seed. So these are some obvious reasons why plants produce colours. And in fact, it's these last two reasons that really illustrate why plants need to be such sophisticated chemists. It's because they're stationary. They can't move, so they develop chemistry to enable them to do things that they would otherwise do with mates that we do. For instance, they have to attract pollinators to be able to transfer pollen from one flower to another. and they have to protect themselves where they stand. And you can see here some of the more obvious examples of protection in plants are thorns and spines and stings. But these are very costly to make, actually, and they don't necessarily always work, because you can see here's a giraffe eating the same bush that this thorn is on. So they often produce chemicals to protect themselves from smaller herbivores and also from diseases. They have some sophisticated mechanisms where they're even able to attract beneficial insects or predators to come and feed on herbivores by sending out volatile chemicals. So they're very sophisticated chemistries. But the one defence mechanism that's probably had the most interest is that of azadirachtin. This is a chemical, a very sophisticated chemical, a diterpenoid from the neem tree, Azadirachta indica. It was so the sayings go, it was discovered because after a swarm of desert locusts, it was the only tree that was left standing, everything else was consumed. And obviously this attracted the attention of some entomologists who carried out some feeding experiments, not unlike the one I've illustrated here, to show that actually azadirachtin was, and still is, one of the most potent deterrents that we know from plants. Just about 1 nanogram per centimetre squared is enough to prevent insects from eating these leaf discs. So some of these mechanisms are very sophisticated, and it's the chemistry that makes them particularly interesting. It's taken Steve Ley, a fantastic chemist at Cambridge, about 25 to 30 years to actually synthesise this molecule. Obviously completely uneconomical, so it can't be used to actually make a product. But he's a very determined guy. But just to give you some indication of how sophisticated plant chemistry is and how sophisticated plants are at making these chemicals. So in my own career, I've tried to look at ways that we can exploit plant chemistry to the benefit of smallholder farmers, and particularly in agriculture and food production. Smallholder farmers are a huge contribution to the health and food of the globe. They provide about 80% of all food in developing countries, so are critical to global health. And they actually employ, at least part of the time, about 25% of the world's population. They're typically very small. In Africa, farm sizes are often less than 1 hectare, producing perhaps less than 1 metric tonne per hectare, to give you an indication of how precarious this existence is. And so anything that we can do that augments or enables these smallholders is going to benefit the globe, hopefully. And of course, insect pests and diseases are a major hurdle for food security in these environments. Insect pests are arguably one of the most important party constraints, particularly for smallholder farmers, not just because they cause a lot of damage, but actually it's quite easy to see them, and it's quite easy to see what damage they do. And so farmers are particularly interested in them. It's not necessarily that they are causing all the damage. Of course, diseases are very important too. But also, the results of effective intervention are very easy to see as well. So it's very easy to bring farmers on board with interventions that actually address insect pests in these farming situations. Synthetic pesticides present an obvious option for controlling insects, but they have limitations in smallholder farming. This photograph I've got of a boy spraying beans in Malawi, I think that is not an untypical sight in Africa. He's obviously not particularly well dressed for the occasion, and also the risk to him is also exacerbated by the risk to consumers too. The impact of these pesticides can be quite serious for the ecosystems as well. We can have predators, parasitoids that may otherwise be consuming pest insects, but also these chemicals can affect pollinators and generally the ecosystem health. Pesticide resistance is becoming more prevalent, so the pesticides that farmers are using are not actually necessarily working as well as they should be. And one thing that's perhaps a little bit peculiar to developing nations, where the infrastructure isn't so good to monitor the use of pesticides, there's quite a lot of adulteration, so farmers may be using diluted products that are never going to deliver what they say they should be on the tin anyway. And of course, finally, they do cost money. And with such a precarious existence for many smallholders, it's quite possible that they can't be afforded. So my career started off looking at exploiting the natural ability of plants to defend themselves against herbivores, to see if we could exploit that as an alternative to using pesticides. So I spent my early career with a former NRI staff member, Dr. David Padgham, looking at host plant resistance in groundnuts, particularly to the Lepidopteran Spodoptera litura. Groundnuts are a major crop in India, and during the 1980s particularly, Squiloptera litura was a particularly widespread pest causing a lot of damage. And so there was a lot of interest in looking for alternative methods to control the insect, particularly as a lot of pesticide resistance was building up. So we looked at resistance in the cultivated varieties, assured there was some. And unfortunately, after my first 6 months of my PhD, I found nothing at all in the cultivated crops that they had at our research farm in India, and left me in a slight state of panic. The reason for this actually is probably because over millennia of selection for uniformity, for yield and other qualities like this, we tend to actually breed out characters like defence. Luckily, at this particular research station, ICRISAT in India, they had a fantastic collection of wild species of groundnuts. And they had this collection purely because they contain potentially useful characters. So I investigated these and continue to do this for the rest of my PhD and found several interesting compounds that are actually not that uncommon in plants. 2 examples here, rutin and chlorogenic acid. But we found these in quite high concentrations in the resistant wild species, and we put them into feeding assays into these artificial diets, where we then allow these insects to feed on the diets that are treated with these chemicals that we've isolated from the plant. And you can see here, I hope you can see these because they're quite small, but this is the insect after 14 days feeding on these diets in the control, so this is untreated, and you can see what effect at the highest concentration of this particular compound that we've got from this wild species of groundnut had on the insects. They simply don't grow. So you can see what an amazing effect this plant compound has on this pest insect, and eventually these insects die. And these compounds are now being used as markers in a selection of groundnuts which are being bred for resistance to these insect pests. So this is now a tool that's being used to develop new varieties of groundnuts. One of the great things about working with Dave Paget is he really encouraged me to throw myself into local cultures, and you can see here some examples of my early days on assignment with NRI in Bhutan and in Rajasthan. It's a shame Dave isn't here today because I'm sure he would find these pictures particularly amusing. Dave actually spent a year and a half in Bhutan, and these are some of the friends he made when he was there. But with Dave, I went on to look at natural resistance in rice, and this was based in Sri Lanka. And actually, NRI spent a lot of time working on brown plant hopper, this particularly important pest of rice in the Far East and also in South Asia. It's a problem because it causes hopper burn when populations get very high, and this is caused by excessive damage from brown plant hopper. And we looked at landraces, which are not wild species, but they're varieties that have been allowed to kind of develop a bit of genetic diversity because they've been bred locally. And they have this genetic diversity that actually provides more opportunity to find things like resistance. And so we looked for resistance in these varieties and found another group of compounds called apigenin glycosides, and this particular example here is shafticide, in the phloem, which is the part of the plant that this insect feeds upon. We found these chemicals in quite high concentrations in the resistant variety. We developed this rather peculiar bioassay where we put a sucrose solution inside a paraffin sachet, and we used that to be able to test chemicals and compare treated sugar solutions against untreated sugar solutions. And you can see here in this graph that in the control, there's quite a steep drop-off in survival of the insects, largely because this isn't exactly an ideal situation in which they grow. But if you add these chemicals from the rice plant to this mix, you can see there's a massive drop-off very rapidly in survival after just a few days. And we found out this because this is a very strong feeding deterrent, so it prevents the insects from feeding. So this is another chemical that can be used as a breeding tool for developing resistance to pests in the field. And this work on resistance continues today, and I still have a project now looking at resistance to weevils in sweet potatoes. This is the main pest of sweet potato, this creature here. It's just a bit less than a centimetre long. Both the adults and the larvae feed on the roots, and you can see the kind of damage that it causes. This reduces the quality and the yield of the potatoes, and this has a serious impact on food security for very poor families in many parts of particularly Central Africa. And of course, because it's a subterranean pest, it's very difficult to control with insecticides. And so actually natural resistance appears to be a very useful way of dealing with this pest. And we did field and laboratory trials to show that the resistance that we had in some of these land races in Uganda was very active. And in fact, some chemical analysis that I've been carrying out and is still going on at the moment, and this is an opportunity for me to say a big thank you to Dudley Farmer, who I've worked with for the last 20 years and has always been a fantastic help on nearly all my projects. We found that the concentration of a group of compounds that were hydroxycinnamic acid esters, particularly this one, caffeic acid esters, this R group here is a long carbon chain, and these occur in the latex of the root, but we found actually that they were in high concentrations on the root surface of the resistant varieties. And of course that's where the adult it's the first point of interaction with the root. And so this is a very important finding, especially since the most popular varieties that are grown actually contain none of these compounds on the surface, and they're highly susceptible to this pest. And another big shout out to David Hall, who has been also a fantastic guide and mentor for me while I've been here. I can't see in the audience at the moment, I'm sure he's here somewhere. There he is. And actually synthesised these compounds in the laboratory, which meant we were able to carry out lots of experiments with them. And what we did with these compounds was we placed the same compound that we found in the resistant varieties on the roots of the susceptible varieties, and then we put the insects on those varieties, and we showed actually that it reduced the feeding holes dramatically compared to the control roots, and reduced the droppings, which meant that the adults were feeding less, but critically, they laid no eggs. And so this is a very interesting chemical on the root surface of these sweet potatoes that may confer resistance to this pest. And it's now part of a breeding program with a US variety, which has other useful qualities like, for instance, orange flesh. Orange flesh is a very important quality of sweet potatoes that's a big part of development programs that NRI is involved in at the moment, because the carotenoids that give it that orange colour are an important precursor to vitamin D. And vitamin D deficiency is a huge problem in children in sub-Saharan Africa. So if we can combine orange flesh sweet potato with resistance to, for instance, sweet potato virus disease and weevil resistance, then hopefully we can get some super clone of sweet potato. But host plant resistance isn't always a good thing, because it may interfere with other biorational pesticides, such as nucleopolyhedrosis virus. And this is some work that I'm undertaking, or have been for the last few years, with Dave Jivich. And it's very nice to see our PhD student, Ali, at the back, who's come along, who's carrying out the work on this subject. So what we find with chickpeas is they produce chemicals such as these organic acids, and they exude them through these hairs on the surface of the leaves and on the beans. And this is actually repellent to Helicobacter pyromigera. Obviously it's not doing a particularly good job here, but they are repellent to insects. And also we find isoflavonoids in chickpeas that confer resistance to fungal wilt and other fungal pathogens. And what we've been finding is that both these isoflavonoids and these organic acids appear to inactivate this virus. So we can't really use these two things together unless we can find a way of preventing this inactivation. So resistance provides a fantastic alternative to pesticides, but we need to be aware that it needs to interact with other appropriate biorational methods of pest management. But it's not just pest management that these chemicals that confer resistance can interfere with. And this is work that I'm currently involved with, we've just done with Dr. Sarah Arnold, who's also here in the audience today. We looked at Lupinus mutabilis, which is actually a cultivated crop in the Andes. It's a very important source of protein. All plant parts contain this alkaloid, D-lupinin, and various other derivatives of lupinin, actually. And lupinin is a fantastic chemical, because in the varieties that they grow in the Andes, the lupins look like this, because this lupinin is a plant defense compound, and it's toxic and repellent to aphids. The ones you grow in your garden have had this lupinin grown out of them, and so they're more likely to look like this, covered in lupins. The problem is, even though this looks ideal, the problem is, Sarah and I found lupinin in the lupin pollen. And so this means that the pollinators, primarily Bombus species, will consume the pollen, and we didn't know what that did. So we set about trying to find out. Actually, we expected it to have some effect on mortality, and to be honest, we were slightly disappointed that it didn't have an effect on their mortality, but I guess it's a pretty good thing. So there was no effect of lupinin in the pollen when we fed it to these micro colonies. But what we did do was show that those colonies that were fed lupinine at high concentrations, this kind of concentration, this is the concentration that we're finding actually in the pollen, produced fewer and smaller males, which may seem like a fairly small effect, but it's a sublethal effect, and sublethal effects can actually mount up and have long-term consequences, but just in their own right, they could have potentially serious consequences for colonies. For instance, sex ratios in bumblebees are known to be important. You do always get more males than females, and this is for a reason. Competition for access to females could be compromised for smaller males. And female choice is important. If males don't cut the mustard, they simply might not want to mate, and that could be important for the following generation. And we know also that small males are poorer competitors. But just imagine if the only food source for males is lupins, and that's the only thing that all the colonies are feeding on. The consequences then for all the colonies could be much more severe. So while natural resistance appears to provide an ideal solution to dealing with crop pests, it doesn't always match up. And one of the things that we're considering at the moment is, could toxic fluorochemicals actually contribute to pollinator decline? And of course this is a very important issue facing agriculture across the world, but particularly in Europe. Pollinators are exposed to multiple stresses in the environment. Several of these have got a lot of press in the last few years. You'll all have heard of the ban on neonicotinoids because there's strong evidence that the consumption of neonicotinoids, even at residue levels in nectar, actually has sublethal effects on bumblebees, and of course this has led to them being banned. But there are other factors as well. The diseases of bees are very important in pollinator decline, colony collapse disorder, habitat loss and land use change, and all these other factors are very important. But actually most of them don't, they have sublethal effects. And so what we'll probably find is it's the interactive effects of these stressors that could put bees at the tipping point and actually lead towards pollinator declines. And in this particular case, I've been looking at plant chemicals and how they potentially interact with some of these other stressors. I'm not going to be able to talk about it today, partly it's very new research, but we're particularly interested, for example, in how plant chemicals interact with bee diseases. They could exacerbate them and make bees much more susceptible to these diseases. Or bees could potentially self-medicate. So there's a potential gold mine of information to still be found out in that area. I'm not going to talk much about it, but I am going to talk about plant chemicals and how they affect bee behaviour, and also how nutrition interacts with other stressors too. Nutritional balance in bees is very important, and this is work that's currently ongoing with Dr. Jerry Wright, she's now Professor Jerry Wright, at Newcastle University. If you cage honeybees in a box like this and you provide them with different sources of food, where the proportion of carbohydrate to protein, by which I mean sucrose to amino acids, is different, you can actually measure how much of each of these they feed upon. And then you can calculate what their intake time is, what the balance between their carbohydrate and proteins. And you find that actually with all bee species it's different. So honeybees, at least foraging honeybees, need a lot of sugar, as you imagine, because they're busy bees. So they have an intake target of about 1 part protein to 250 parts carbohydrate. If you take bumblebee workers, they're also pretty busy, but their intake target is about 1 part protein to 90 parts carbohydrate. And even within the same species, you can see honeybee foragers and honeybee nurses. Honeybee nurses obviously are not quite as busy, well, not flying about as much. Their intake target is 1 to 50. So this is very important because actually what most bees are presented with in our agricultural ecosystems is a pretty uniform plantation of a single crop species. Now this one crop species can't possibly be delivering the intake target for all the species of bee that occupy our environments. It's not just honeybees and bumblebees. There are hundreds of species of bee that need to have their needs satisfied. And of course, the landscape that they've come from is probably more like this. So bees need a mixed diet just like we do. And of course, if we have that mixed agro-ecosystem, we can also enhance natural enemies, because natural enemies also need nectar and pollen, and will probably also have specific nutritional intake targets. So we do need to really consider this particular fact about these about nutrition. But there are some very interesting things about the chemistry of nectar And we're currently working on several of these at the moment. For instance, we found this particular diterpenoid, granutoxin, in the nectar of rhododendrons. Actually, we've known it's been there probably for centuries because it's used to produce a rhododendron honey by a specific honeybee subspecies in the Caucasus, Apis mellifera caucasica. In fact, it's called mad honey because it has these particularly weird effects on humans. It makes them dizzy, causes low blood pressure, slows the heartbeat, and in fact, in high doses, it can even actually stop the heart altogether. I don't know whether any of you recall the Hollywood remake of the movie Sherlock Holmes, and the arch baddie Lord Blackwood actually took Rhododendron ponticum to make himself look like he was dead. Dr. Watson pronounced him dead because, of course, he couldn't feel his heartbeat or his pulse, and they actually got the species name right and everything. And in fact, rhododendron honey has been used as a weapon of war. Mithridates VI of Pontus, who considered himself an arch enemy of Rome, he was probably just one of many enemies of Rome, considered himself an arch enemy of Rome, was quite a fan of plant poisons, and his army left honeycombs of toxic honey on the road which they knew Pompey the Great's army was marching down. And of course, after a long day march, this army couldn't resist these honeycombs, so consumed them, and of course were left in a complete stupor and nausea. And then during the night, Pontus's army allegedly came in and slaughtered them in their beds. So an interesting use as a weapon of war of plant chemicals. But on a more serious note, of course, rhododendron is a very important invasive weed in the British Isles. So we're interested in finding out what it does to our native bees. This is the native range around the Black Sea, and in fact there's also some the native range on the Iberian Peninsula. This is its invasive range. You can see the British Isles is completely covered, and there's a big area of risk in France as well. So is rhododendron nectar toxic to pollinators? Well, we've carried out some experiments in collaboration with Trinity College in Dublin. This is the work of Erin Jo, who's a PhD student there, and she's shown that honeybees that are made to feed on granutoxins, these are local honeybees from the British Isles that are made to feed on granutoxins or the rhododendron nectar. This is the artificial nectar here. This is the rhododendron ponticum nectar, and this is the artificial nectar with granutoxins at around about the concentration we find in nectar. And you can see in a matter of hours, our native honeybees die off. So clearly, the honeybees around the Black Sea have adapted to this compound and potentially use it for some reason, we don't know what. But here in the British Isles, this is a serious problem. If we do the same experiment with Bombus terrestris, which is the bee you're more likely to see feeding on rhododendron, you'll find even after 30 days there's no effect at all. There's no difference between the artificial nectar and the control. Even with a mining bee species, Andrena carantonica, we find that there's no difference in survival. So for honeybees, it's toxic. It kills them pretty rapidly. For these other two species of bees, they seem to be fine. But are there any sublethal effects? Well, if you feed Andrena carantonica with granutoxins, we find that actually it doesn't spend any time flying, which will make it, one, not a very good pollinator, and two, not a very good forager. So actually, this sublethal effect could have a serious consequence for the colony. And if we do the same experiments with Bombus terrestris, we find there are no effects of this toxin on Bombus terrestris. And in fact, Bombus terrestris shows no excessive grooming or paralysis, which are symptomatic behaviours associated with toxicity, whereas Andrena carantonica spends 20% of its time trying to clean itself or lying on its back, not able to do anything. So the consequences of granutoxins are quite severe for some species of bees, but not all. And interestingly, we find that actually the concentration of granutoxin in the nectar is lower in closed flowers than it is in open flowers, suggesting there's some expression of this chemical in the nectar as the flower opens. And this is some work that we're actually doing on Arabidopsis, looking at expression of compounds in open and closed flowers currently at the moment with Eleanor Thompson, who unfortunately can't be here today. So is this something that's being done effectively by the plant to garner some kind of pollinator fidelity or specialisation? One bee's food is another bee's poison, because if we look at areas in Ireland where rhododendron is absent, we have about 30 colonies of bumblebees per hectare, but where rhododendron is present, we have about twice as many colonies. So it's kind of interesting that these compounds appear to favour some bees and they appear to disfavour others. And this is important in specialisation. Pollination is a mutualism that exchanges food for sex. I'm afraid this is about as sexy as the talk's going to get. I know all the EMR people have turned up for the sex and the drugs. Pollinator loyalty is obviously a very important part of this process because it's up to the plant to ensure that its pollen can reach a conspecific flower. With generalists, the pollen could go anywhere. But by specialising your pollinators, you can increase the chances of the pollen reaching a conspecific flower. And it's up to the plant to generate this pollinator loyalty. So toxicity to all but a few species of bee, for instance in rhododendron, increases pollination success. But bees also learn to choose flowers, particularly those that provide a good reward. And they also, particularly honeybees, have this interesting behaviour called floral constancy. And this is kind of temporary specialisation on a single species. And this might be even in the presence of another species that's producing plenty of food as well. There's a great benefit to the flower, of course, because this increases the chance of the pollen transferring to the conspecific. But for the bees, there could also be benefits. It might be a more reliable source of food. The scout's gone out and located a good source of food and it's returned to the hive, so all the others are going to go there. It might also reduce competition. So there are potential benefits of this. But what actually drives floral constancy in honeybees? Well, it's actually simply memory. It's memory of a reward associated with a cue. So a flower odour, a colour, a shape, a landing pad. You've all seen on digitalis, there's a landing pad they have. And so bees make these strong memory associations. I'd like to point out one thing here. And just as they're landing, you can see this one's received a cue that it's about to feed because it's got its proboscis sticking out already. I'll draw your attention to this again in a minute because this is an interesting bioassay that we do that involves that. So this brings me on to the last main story of the talk, which is about caffeine. You may be surprised, or maybe not surprised, to find that actually caffeine occurs in coffee, but you may be a little bit more surprised to find that actually caffeine occurs in the nectar of coffee. It also occurs in the nectar of citrus, and this is work that I've been doing again with Jerry Wright at Newcastle. You might notice here that she's wearing a bee vest. It's a curious thing, actually. Apparently you smother yourself in Vaseline that contains queen pheromone, and then you smear your arms with insect repellent where you don't want to go, and then you walk up to the hive, and the beekeeper opens it up and they just cover you. I mean, it's just a bizarre thing. In fact, Sarah was telling me you can even get bumblebees if you're very clever. Anyway, so I've been doing some very interesting work with Jerry on some interesting behavioural effects of caffeine. So yeah, where are we? Oh yes, we're finding caffeine in the nectar of citrus and coffee. In fact, caffeine occurs in about 13 genera. About 100 species of plants we know at least contain caffeine. And so we found it in the nectar of these species. And the concentration is generally pretty low, but interesting, it's around about the same concentration as you'd get in a cup of Nescafe, at least in the nectar of Coffea canephora, which is this one here. But interestingly, Coffea canephora, which makes the cheaper branded coffees, the instant coffees, is an obligate outcrosser, and it has more of this caffeine in it than Coffea arabica, which can self. And that will become more interesting later, and I'll come back to that little topic. Basically, all the species that we looked at in citrus and coffee contain caffeine. So why does it contain caffeine? Well, the puzzling thing for us initially was the fact that caffeine is a toxin. It's actually a feeding deterrent. That's why the plant produces it. That's why you get so much of it in beans. That's why you get so much of it in tea leaves, young tea leaves, because it's being produced to protect the plant from herbivores. And you can see here from Liz Bernays' work from about 15 years ago, where she showed as you increase the concentration of caffeine in the feeding substrate, these two test species reduce their feeding. So it's a feeding deterrent and it's a toxin. And actually, with honeybees, it's also a feeding deterrent. So we were very puzzled indeed. As you increase the concentration of caffeine, the feeding deterrent response increases and even is detected in antennae at the highest concentrations. But in the nectar, the highest concentration is here, about 0.1 millimolar. And the honeybees' taste detection threshold is actually too high to detect caffeine in the nectar of coffee and citrus. So it must be there for another reason. And we know that caffeine has interesting effects in other animals. We know, for instance, that caffeine improves memory in rodents. So we wondered whether caffeine could influence bees' memories the floral traits associated with food. And of course, there's this very interesting biomass that some of you may have come across called the proboscis extension reflex. And you may recall that I pointed out this behaviour here. As the bee is approaching this flower, it recognises either an odour or a colour or a smell or a shape. And as it's approaching, it's getting ready to feed because it's received that cue, so it's extending into the proboscis. It's remembered that cue. And we can do a similar thing and test memory in the laboratory. What you'll see in this video here, Dr. Jerry Wright is providing this bee with a sugar solution. And at the same time, that insect is getting a small plume of an odour, in this particular case 1-hexanol, which is a generic green leaf volatile. So it's able to associate that smell with that food. And we have this process where we have 6 conditioning trials with a short interval in between them. And the insects, as it says, are rewarded with the sucrose solution. And then after 10 minutes, we can do experiments a little like Pavlov's dog. You recall Pavlov's dog salivated when it heard the bell, because it associated the bell with the food. Well, we have a similar experiment here. So in this video, you'll see, obviously we can't show you the odour, but when the red square comes in, that's when the odour plume comes in. So the odour plume will come in for 4 seconds, and the insect extends its proboscis, expecting to feed, but of course there's no food. And so we're able to show that that bee is simply extending its proboscis because it's remembered that cue. So what we've done is we've done experiments where we've tested this over 10 minutes, short-term memory, which actually we expect bees to have, and also long-term memory, 24 hours. And do excuse the extent of this histogram, but I'll try and explain my way through it. What we found was, as you increase the concentration of caffeine in the food that we're giving to the bees, after 10 minutes, the proportion of insects that are remembering the flower cue are not different at all. So after 10 minutes, caffeine doesn't appear to have any effect on the memory at all. But if we test those bees after 24 hours, you can see that in the control, the proportion of bees able to remember that odour has dropped right off. And as you increase the concentration of caffeine in the nectar, you can see that more and more bees are able to remember that cue. And this hashed area shows the concentrations which we find in the nectar of coffee and citrus. So what we propose is that caffeine enables bees to be better able to remember flower cues. And if they're remembering flower cues for coffee, it means the following day they're more likely to visit those plants first because they already have that memory of that association with a food reward. And what happens in this situation, we've done some experiments, Mary Palmer at least has done some experiments in Dundee looking at what caffeine does to the bee's brain. Kenyan cells, these sensory neurons in the mushroom body of the bee's brain, and what caffeine does, it depolarises these the membrane potential of these Kenyan cells, it effectively increases the excitability and so makes them more likely to fire. And they're very important in the formation of memory. And so if they're more likely to fire, they're more likely to respond to these cues associating flower odours with food. And so we actually have pharmacological evidence that there is an effect of caffeine on the brains of bees, making them better able to remember these flower cues. So what does this mean for the plant and for the pollinator? Well, for the plant, of course, this improves floral constancy. Bees, as I say, are more likely to continue foraging on that species, and they're more likely to transfer pollen from one flower of the same species to another. So this is an evolutionary adaptation. With pollinators, of course, bees are more likely to recruit nestmates, and so they may have more secure sources of food because it's a more reliable source of food. The scouts are spending less time in the morning looking because they already have that cue in their memory. So there are benefits for the plant and benefits for the bee. So what about for coffee and for us, and for citrus, of course? Well, coffee is the second most traded commodity on the planet after oil, so almost anything you can find out about coffee could potentially have a huge impact on an enormously important commodity. We know that enhanced pollination in coffee improves yield and quality of that coffee. So one thing we're thinking possibly is could we use selection of provenances when replanting to ensure that those provenances have caffeine in the nectar. You'll recall that Coffea cannifera was the self-incompatible species, so we must have transfer of pollen from one flower to another. And this was the species that had the highest concentration but still below the bee's taste threshold. In citrus, there are lots of different ways we could potentially use this. Pollination reduces a fruit drop, and this is particularly important, of course, in drought conditions. But in satsumas, because we're unable to, at least some of us are unable to consume fruit that contains pips in it for some reason, so farmers need to produce satsumas. Could we deselect for caffeine in provenances of satsuma, potentially? And in any event, pollination improves yield in ecology, so it could be useful there too. Could we use this information to manipulate other species of bee that are important in food production and encourage a sort of forage focus? In soft fruit, for instance, particularly strawberries, pollination by bees is very important. Insufficient pollination results in deformed fruit, and of course farmers find it very difficult to sell these. And so what we're considering at the moment, Sarah and I have got a proposal at the moment, it might sound a bit wacky, but we believe in it, is that we might be able to actually improve the ability of commercial Bombus terrestris to pollinate strawberries. Farmers actually spend tens of thousands of pounds a year on commercial bumblebee colonies, which is extraordinary, really, because wild pollinators are not doing enough of a job. And these bees could be distracted by heterospecies. They could also be actually feeding on those resources that might otherwise be used by native or wild pollinators. So, and I understand the motivation can sometimes be low when you've been transported across the channel from the Czech Republic or somewhere, and so we might even be able to increase the motivation of these bees by training them to recognise flower odours of strawberries. But we'll see how this work develops. But looking forward, at least from my own perspective, increasing food production obviously is a very huge task with an increasing population and reducing land mass. Anything that we can do to improve food production is going to be important. And we have a particularly strong focus in our life at the moment on sustainable agricultural intensification, simply because there's little room for land expansion. And I think one area that I'm particularly interested in getting more involved in is exploiting this natural capital, in enhancing the agricultural ecosystems, potentially augmenting beneficial insects, both the predators and parasitoids and pests, but also pollinators, and also potentially exploiting wild plants as pesticides. In fact, this is an area of work that actually has occupied quite a lot of my time over the last few years, but I haven't yet had a chance to mention. So I will very briefly now talk about work that I've been doing with Steve Belnane over the last probably 10 years, I think, isn't it, Steve? Where we're looking at trying to optimise the use of plant materials as an alternative to pesticides. When we talk about pesticide in plants, I don't mean botanical insecticides, the kind of thing that's a plant derivative that you might buy on a shelf in a bottle. We're talking about really unprocessed plant materials that require fairly rudimentary preparation, so they're actually very well suited to smallholder farming. The pesticide efficacy of these plant materials relies upon this natural ability of plants to defend themselves against herbivores, because the wild plants, they haven't had these qualities bred out of them. And some plants have very potent efficacy. They're obviously low cost, they don't cost anything but the time it takes to harvest them and prepare them. They're also familiar to farmers. In fact, most of the species we work on, we found out about them by talking to farmers. And they said, oh, this is what my mother or my father used to use, or my grandfather or whoever used to use, and then we tried to find out more about them and optimise how they're used. And they're also more available when required. As I mentioned earlier, as I might not have mentioned earlier, one of the problems with synthetic pesticides is everybody wants them at the same time, because you might get an invasion, and of course you'll never have a supply. And of course the more remote farmers are the ones that are more likely to miss out. So if they already have an alternative, then this may enable them to have more food security. So just as an illustrative example, I can't possibly start going into this whole subject area, but just to kind of illustrate what we can possibly do with pesticide and plants, I can illustrate it here with some Ghanaian plants that I've been working with as a biorational pesticide. This is on cabbage. And you can see here what happens if you don't spray, or if you just spray water. And this is damage initially caused by the cabbage aphid, Brevicarium brassicae, and then later exacerbated by the diamondback moth, Gutellus ardestella. So you can see not very good yield. And of course this is what you get with the conventional insecticide, at least one that works. So I can tell you now that we have another insecticide in this trial that looked a bit like this. so they don't all work. But this one did work. I'm not going to mention his name because I'm going to hurt. But when we used this wild species that grows on farm borders and all around the farms, we made an extract of this, about 5%, and sprayed it. We got just as good an effect as we got with the pesticide. But the great thing about these plants is their effect is quite short-lived, and so they don't have the same persistence as some of the synthetic pesticides, and they don't have the wide or broad efficacy that some of the synthetic pesticides have. And so we monitored beneficial insects in the same plots after we actually sprayed, and we can show here that I'm telling you what that pesticide is now. It's attack. But now you can see what it does. So in plots where we use attack, we have very low numbers of beneficial insects, hoverflies and ladybirds and spiders, which of course are feeding on the pest insects, the aphids and the Lepidoptera. But even though we had lower numbers generally in the wild and cultivated species of plants that we used as extracts, they were all much better than the synthetic pesticide. So this just illustrates a little bit about the potential of these plant biopesticides. We've also done some work that shows that they are economically as viable, if not more viable, than the synthetic pesticides, if you calculate the amount of time invested in making the extract against the cost of the pesticides that you buy in the shop. So I'll leave you with this thought about how I would like to spend the rest of my career exploiting the natural capital that we have at our disposal, and consider that there's actually quite a complex ecosystem that we have the benefit to exploit. We have our primary crop, of course. This is a slide I pinched off Jeff Gurr. Stephen recognised this from about a month or two ago. But I think it illustrates the potential for us to exploit ecosystems very nicely. We have this primary crop, in this particular case rice. We have arguably our secondary crop, because it could actually produce grain or vegetables, but it could also provide a source of botanical insecticides. I've just illustrated the potential benefits of those. But it also is a habitat for natural enemies and pollinators. And we need to enhance these environments and make sure that we can engineer them so that they enhance the beneficial insects and not necessarily provide habitats for pest insects too. But of course we have a tertiary crop too. We have another level where we can have timber or fruit, we can have drought fodder. So we need to think of the whole farming system as contributing in many ways. And of course, once the whole ecosystem is taken care of, then there are enormous benefits in many other different facets of agriculture and farming. So finally, it falls to thank all the people that have been involved in the work that I've been involved with. I'm very glad to see Nigel Beach has come from Kew Gardens today, from the Sustainable Uses Group. Nigel Beach is a natural product chemist extraordinaire, and he's particularly expert at nuclear-magnetic resonance spectrometry, which is an essential technique we have for elucidating the structures of the chemicals. A lot of the chemistry you have to take for granted in this talk. Well, I'd like to extend my thanks to Nigel not only for 20 odd years of working together, but also for coming today. But also Wally Sims, who's a great collaborator, long-term collaborator of NRI, and other colleagues, Jeff Carter, Porter, and Ian Farrell at Kew Gardens. And also, I'd of course like to thank all my colleagues here at NRI who haven't yet been mentioned, particularly all the staff that never get a mention. Thanks, Heather, for organising today, and to Mark Parnell at the back for making almost everything else possible with all my projects. And finally, of course, I'd like to thank the Stevensons, who put up with an awful lot of my travelling overseas and somehow managed to cope with it, despite he's too busy on my iPhone. And also, of course, I'd like to thank you all for making the effort to come and listen to my story today. So thank you all very much for your attention.